Okay, just to get started here, again, welcome everyone to our 2021 Michigan Sea Grant Undergraduate Environmental Internship Symposium, when all of our 13 interns are going to be able to share what they've been doing this summer, which are some really great projects. Before we begin, I was wondering if we don't really have time for everyone to do introductions of all the nearly 60 people that are attending. So we would love it if you would put your name, where you're from, and your email address in the chat. This will help us know who's attended the symposium, but also we would love to send you a feedback form about the symposium so you can give us your feedback about how we can make the symposium work better, even better next year. So if you could do that in your chat, and Emily Rao from Michigan Sea Grant, she's an employee of Michigan Sea Grant. She is going to keep track of the chat, and she'll be recording that information that you're sharing. Other people, please look at the chat and see who all is here. It's our way of introducing ourselves to each other. So if you want to say a little bit about yourself besides where you're from, please do that as well. I'll share the agenda in a few moments, but we will have a few opportunities for all of you to ask questions of the interns and their projects. We have 3 different time frames for you to ask questions. When that comes about, you can either use the chat or you can use the raise your hand feature in Zoom to ask your questions in person. Again, I mentioned that the meeting is being recorded, and we'll be posting that on the Michigan Sea Grant website under the internship page. You want to go back and look at that in the future. And I think everyone already knows is well familiar with doing Zoom meetings. And so please do mute yourself unless you're speaking, and that will enable us all to be able to hear the presenters better. So those are kind of the rules of the road and introductions to get started. If anyone has a question along the way, please do put it in the chat. Emily will keep track of that and let me know, because I may not be able to pay attention to the chat. I wanted to start off, just because there's so many people here that may not know, just a little bit of information about Michigan Sea Grant. So Michigan Sea Grant is one of 36 sea grants around the country as a part of the National Sea Grant College Program that's funded through the National Oceanic and Atmospheric Administration. It's a collaborative program of the University of Michigan and Michigan State University. At the University of Michigan, we have the main administration. We run the research program and the communications programs out of the Ann Arbor office. And then at the Michigan Sea Grant, at the Michigan State is where we have the administration and of the extension program that's a big part of Michigan Sea Grant. The mission of Michigan Sea Grant is that we support research, education, and outreach projects that serve to enhance the sustainable use of the Great Lakes resources that benefit local, state, and national economies. We use this, our vision is to use these research, education, and outreach efforts to maintain a healthy and sustainable Great Lakes resources. And we achieve that through integrating with our universities and public and private sectors. So just a little bit about the 3 different main programs that we have. The research program, again, which is out of the Ann Arbor office. We support university research investigators to do research projects. We support graduate fellows, and they have a fellowship. We run these in research projects and graduate fellowships on 2-year cycles. And then in the past 3 years, we've had this undergraduate internship that's also a part of our research program. And those are on annual cycles, so every year. And all of these research projects the intent is to support applied research and engagement with our communities. Our research projects, they are intended to develop science-based information and tools that we can use to solve problems in our communities and inform decision about complex social and coastal issues. At the MSU office, we have the extension program, and those extension educators, they live and they work in coastal communities. You'll see pictures of them in a bit and what communities that they work with. And they help apply the research, and they conduct educational activities and work with stakeholders around the state. to help solve problems and convey information about these complex research outcomes. And then the communication programs is led through the Ann Arbor office, but also has a representative in East Lansing office. And they work with extension educators in the research program to develop outreach and educational products and services. And they also do our Michigan Sea Grant newsletter upwellings and various social media blogs and posts. And our interns have contributed some of those over the past summer. And we hope, again, even more coming forward. Just to show you a little bit about who we are, the people who have been, you've been contacted, been working with the interns, it's myself. I'm the research program manager and assistant director of Michigan Sea Grant since 2011. And this is shortly to be my last month. I'm retiring. And so the person who will be taking on the research program manager position is Mike Fraker. He started in August and will be the research he will be managing the internship program going forward. Emily Rao, she's a research program assistant for this summer. She's been really a great help and has been the person contacting the internships most often. Following this internship, she's going to be going on to have a research, a fellowship with the Wisconsin Sea Grant, working with her coastal zone management program. So we're excited for her and her next career opportunity. The Administration, Communication, and Research Ann Arbor office, Tom Johangen is our director. Elizabeth Striano manages and leads the communication program, and that includes the Geneva Langland, who is the editor, and Todd Marcy, who is our graphic designer and photographer. Our financial part of the program is led by Elise Larson, and Brandon Euclid is our new administration project coordinator. He's been with our program for just a few months. And then we also have the Great Lakes Aquatic Nuisance and Invasive Species Project that's out of our office. Well, that's led by a couple of different people. There's both out of the extension and out of our Ann Arbor office, but we have two staff people here, Elle Lower and Austin Bartos, that are research associates with that program. And then out of our extension, East Lansing office, our associate director is Bill Taylor, and the person who leads the extension program is Heather Kiesenberg. They have offices in East Lansing, and they lead that program. Also in East Lansing office is Cindy Hudson. She's our other communications staff person. And then Vanessa Pollock, she helps with the fiscal officer for the MSU part of it. And then this is a picture of all of our 12 extension folks that are located all around the state and work with the communities. You can see the different locations where they work with those communities. They have topic areas where they are really specialized in, and then they also work specifically with the communities where they're located. So they really provide a great opportunity for us to have outreach and education in our coastal communities. So then just a little bit about the environmental internship program. Again, I said that this has been in place for this is our third year. It funds undergraduate students from any accredited colleges or university in Michigan. And they work with private businesses, state, federal agencies, nonprofits, or universal faculty to plan or implement an environmental stewardship project in the Great Lakes region. One thing I wanted to mention is that for this past year, for the first time, we actually had 3 different sponsors that supported 6 of our environmental internships, and that we had 2 sponsors from the Michigan Department of Natural Resources, David Clapp and Todd Wills, and they supported 3 students that helped out with their smallmouth bass program. Mike Reed from the Detroit Zoo, he supported students working with environmental education with communities in Detroit. And then David Misford with the Herpetological Resource Management group that had this one student that helped with herpetological habitat assessment. And I really want to take this moment to thank them on behalf of Michigan Sea Grant for providing these opportunities to advance these students' education and provide them with opportunities to work on projects that make such a difference for these agencies and communities. It was really great, and I wanted to thank them for that. The goal for this internship is really to reach a broad, diverse group of undergraduate students and to implement these stewardship projects and provide some professional development opportunities also. So we're just about ready to get started here. This is just the agenda for the rest of the day. You can see that we've grouped our interns in 3 different groups. And so we have presentations about fisheries first, and there's 5 students that will have presentations that are loosely focused around fisheries. Then we'll provide an opportunity for 5 minutes for people to ask them questions. And then there'll be another set talking about sort of field surveys, information, kind of environmental education opportunities, 5 students there. Then we'll have a 5-minute break for everyone to get up and get a drink of water or whatever they need. We'll come back. We'll have 3 more presentations, followed by another Q&A session. And then we'll have an open Q&A session for all of the students. Each student will have about 5 minutes for their presentation. They all are aware of this, and I'm sure they've all practiced. But in case they go a little bit over, Emily will be keeping track of time and let them know if they are getting substantially over their 5 minutes. Any questions about this before we get started from anyone, or any concerns? Let those know in the chat. Okay, we'll get started here. First up is Rachel London, and she's one of the people working with the MDNR on the smallmouth bass program. And her advisors were David Clapp and Tom Locke. David Clapp was from the DNR and Tom Locke from MSU. Rachel, I forgot to mention before we go is that we pulled all the slides together in one program. So I'm kind of running it. So then each student needs to tell me when to advance their slides. Okay, thank you.
All right, hi everybody. I hope everybody's doing well. My name is Rachel London, and I'm a laboratory assistant at the MSU Aquatic Animal Health Lab, studying environmental biology and zoology. This summer, I've been fortunate to lead a novel collaborative study on the potential causes of smallmouth bass skin lesions. I'm so excited to be able to share this project with you all today, as well as some exciting results. Smallmouth bass are vital to Michigan and our anglers, so reports of skin lesions, such as this one, gain a lot of attention very quickly. Catherine, if you could go on the next one. First, I want to provide you with some background on our efforts. Anglers seeing these lesions on their catch told clerks at the DNR, and soon field teams from other management agencies began seeing similar lesions while conducting field work. Through collaboration between the MDNR and other entities, these reports made their way to my advisor, Dr. Thomas Locke. Soon after, Dr. Locke and I found an exciting opportunity through Michigan Sea Grant and subsequently the sponsor position by the MDNR, and we agreed we had both a problem and the means to begin solving it. This has been a collaboration of individuals to kickstart a solution to an emerging problem in their communities, and it has been truly rewarding to be a part of this effort. Next, please. To sample fish from a range of locations across Michigan, depicted here with these logos, I coordinated with both the MDNR and Sioux Tribe. I am beyond thankful for the individuals and their teams who patiently taught me how to fish. Whether that be by simple hook and line, trapping gill nets, or boom shocking, these teams gave me knowledge in a variety of field techniques, and I was able to successfully catch tons of fish, including the 21 smallmouth bass and 1 largemouth with skin lesions that we have entered into the study so far. Next, please. After collecting fish in the field, I traveled back to the lab where we performed full exams on using a multifaceted approach to collect tissue for a range of analyses. We are analyzing the tissues for parasites, bacteria, and viruses using a variety of laboratory techniques to determine if 1 or more fish pathogens could be potentially causing these lesions. Next, please. We studied fish with a range of lesions, some deep into the musculature with severe hemorrhage, as you see in these photos. We collected tissue from the lesions for bacterial culture and found several bacteria, including this yellow pigmented bacterium seen here on the bacterial plate media. Based on the culture morphology you see in the middle picture, we suspected the isolate to be a bacterium that causes a serious disease in fish known as Flavobacterium columnare. When we ran PCR analysis, we confirmed the identity of the isolate as Flavobacterium columnare, the causative agent of columnaris disease. Next, please. Similarly, we analyzed the tissue for viruses that infect fish. This is a different process than bacteriology, as we infect living fish cells looking for evidence of a replicating agent. To show you what we look for, you can reference the top photo showing unaffected cells, and then reference the bottom photo depicting cell lysis or breakdown. The difference between these two photos gives us reason to suspect an active infectious virus recovered from fish with these lesions. Given the gross appearance of the lesion and the species I'm working with, we did initially suspect that this virus could be largemouth bass virus. Using PCR again, we confirm this to be true. Next, please. Analyses like virology can take up to a month to complete from the day that we are able to place samples on cells. So we are still in the process of finalizing many of these analyses, but I am thrilled to share with you my results to date. As you'll see in this table, we recovered Flavobacterium from skin lesions collected from 3 out of the 4 sites. As I said earlier, this is a bacterium known to cause infection and disease in multiple species of Great Lakes fish. The widespread distribution of this bacterium associated with these lesions is of note. Thus far, we recovered largemouth bass virus from 3 fish with skin lesions at 1 of the 4 sites. As our analyses are still ongoing, it is possible that largemouth bass virus is present at other sites. Largemouth bass virus is most known for the cause of mortality events in largemouth bass. However, recent studies, along with my own, are showing largemouth bass virus can cause disease and possibly lesions in smallmouth bass. Although analyses are still ongoing, these results to date revealed 2 well-known fish pathogens were associated with some smallmouth bass skin lesions in several locations in Michigan. Of course, this was just the beginning, but it is an exciting start that I am proud to be a part of. Next, please. I'm so fortunate for the opportunities that I've been afforded this summer. I've always dreamed of becoming a veterinarian. However, I always imagined myself working with cute, fluffy puppies and kittens. Since this internship, I've completely changed my vision for myself. I want to be in the field, working with fish and wildlife, studying animal health alongside respected, knowledgeable individuals who strive to do their best for the benefit of animals. Essentially, I never want the experiences I've had over the course of this internship to end. Next, please. With that, I'd love to thank the organization that made these experiences possible, Michigan Sea Grant, as well as all of the other individuals and organizations that have supported, mentored, and educated me this summer. Thank you.
Thank you, Rachel. Very nice presentation. Next up is Michaela Kindler. She's from Kalamazoo College, and her advisor is David Clapp, and she's looking at round goby.
Hi, everyone. I'm Michaela Kindler, and my project was on estimating round goby abundance and density in Lake Michigan with photo surveillance, working with my project advisor, Dave Clapp, and other biologists at the Fisheries Division of the MDNR. Next slide, please. The idea for this project initially came to light because of a CMU grad student who has also been doing research with the MDNR on smallmouth bass diets and the role of round gobies in them by using bioenergetics modeling. This grad student, Matt, specifically aims to assess changes in the composition of smallmouth bass diets and the potential predation pressure this Lake Michigan native could inflict on 2 invasive species, rusty crayfish and round gobies. However, to do this, the model requires an abundance approximation of these species populations, so he can calculate the proportion of prey consumed by smallmouth bass versus what is available to them in the environment. This is one place where my research using GoPro photo quadrats finds use. However, an index for round goby abundance in the Great Lakes has been a concern for much longer. A current project on which the Fisheries Division is working, partnered with the Nature Conservancy, is a reef restoration project focused on reassessing the current knowledge on Lake Michigan reefs and evaluating the ecosystem health, like spawning success of native Great Lake fishes like lake whitefish and lake trout. This includes research into round goby populations whose ideal habitat is found in these areas and whose diet consists of these fish's eggs. Quantifying round goby populations in Lake Michigan is essential in determining a successful control method, in addition to ensuring that the management efforts taken for these Great Lake native fishes are the best for their population's restoration. This summer, I've used the PhotoLapse feature on GoPros to capture and estimate an index for round goby's population in East Grand Traverse Bay. The GoPros are attached to baited quad pods baited with dog food, as seen in this picture on the right here, and programmed to take a photo of a quadrat every minute. Next slide, please. And here is an example on the left of what one of those photos may look like. This method has been used before. However, the accuracy of these population indices in relation to round goby's true density is a concern, as what one may see in a series of photos may not fully capture the fish in the area, who may be underneath the substrate or just out of the camera's field of view. The second component of my project this summer has aimed at assessing this, to standardize or ground truth these quantifications from the field. To do this, myself and others worked to transform these 8-foot in diameter grain tanks into large mesocosms, where we could essentially use the same process from the field with a now known density of gobies. We used 2 different substrate types, cobble and sand, and tested 3 different densities, a low, medium, and high, both with baited and unbaited GoPros. The cobble used in the mesocosms was chosen to mimic conditions from the field, as it was used in a previous project to restore a reef in Elk Rapids in 2015. With these photos, I will be able to take the round goby indices found from the field and calculate a potentially more accurate index for round goby's true abundance in East Grand Traverse Bay. This will not only provide Matt with a number he can use in his model, but it will also provide those working to restore the Lake Michigan reefs a better understanding of how round goby populations may actually look. Next slide, please. Although my research is not yet completed, another part of the tanks has involved a similar standardizing approach, but with minnow traps. To gather all the gobies used in each trial, we drain the tanks and basically start our scavenger hunt. However, before doing this, we'll put a baited minnow trap into each tank, leave it for an hour, come back and check to see how many are in each trap. The results of this have been pretty eye-opening. As you can see in the graph on the left, the sand is represented by the blue and the cobble by the red at each density. In the sand tanks, we have been seeing capture rates shown on the y-axis that are more than double those found in the cobble tanks. If these patterns hold, the number of round gobies in the lake may be greater than previously estimated. This may impede current and future restoration efforts and highlights the necessity for more research to be done establishing an accurate method to approximate round goby densities in areas of Lake Michigan. I'm excited to continue this work and to see the results of the research. It's been such a rewarding experience for which I'm really beyond grateful for. Thank you.
Catherine, you're on mute.
Thank you. Thank you, Michaela. That was a really interesting presentation. Really nice job. Good work. All right. Up next is our third student that was working with the DNR, Cheyenne Wilcox, and she's with Oakland University, also working on smallmouth bass.
Hi, my name is Cheyenne Wilcox, and like Catherine said, I'm a senior at Oakland University, and my project this summer was a comparative analysis of recapture data for smallmouth bass in Lake St. Clair and Lake Michigan with the MDNR. And my advisors were Dave Clapp and Todd Wills of the Charlevoix fishery and the Lake St. Clair fishery, respectively. Next slide, please. All right, starting up with some quick background information. Smallmouth bass are a popular game fish throughout Michigan and play an influential part in the reef communities. In order to study them, a process known as jaw tagging allows the DNR to mark bass that have been caught in order to identify them if they ever caught again. Each jaw tag has a unique number assigned to it for identification and data collection purposes. Jaw tags have been used in order to study not only the movement patterns of these fish, but they also allowed the DNR to make population estimates for the species. This process has been occurring for the last 50 to 60 years, but the current staff at the DNR has been more directly involved for the last 15 to 20. In the bottom left picture, you can see an example of what a jaw tag looks like on a bass, and on the right is an example of a DNR staff buoy that marks the location of a net used for fish surveys. Next slide, please. For the methods, our data source primarily consisted of relying on anglers to report any tagged fish that they caught, as well as identifying any recaptures we caught in our nets out on the lakes. There were a few different varieties of nets that we used, mainly what are called trap nets and mini flag nets, which are a little more complex with several wings designed to spread out over a larger area, and also gill nets, which are more simple in design, consisting of one long nylon mesh strand several hundred feet long. The top right and bottom right picture shows a DNR employee, John, on the right, and CMU grad student, Matt, on the left, pulling in a gill net on Grand Traverse Bay. And on top of tagging fish, we also collected data on their lengths, weights, and sex when applicable. Bottom left shows me measuring a smallmouth bass fry that we caught in Lake St. Clair, and bottom center is me measuring a rock bass just for practice. After collecting our data, I then compiled the lists into an Excel spreadsheet, as well as utilize the Access database that the DNR already had to begin making my data summaries from which my graph was made. For my project purposes, I decided to focus on bass that have been recaptured more than once in order to study their movement patterns. Next slide, please. And on this slide are just a few more fun pictures I gathered over the summer that help show some of the other projects I played a part in out on the lakes. The grad student, Matt, is studying smallmouth diets. So the top left picture is me assisting him as he flushes the stomach of a smallmouth we caught in Grand Traverse Bay. And the bottom left, center, and right photos are of me measuring and holding fish from a nearshore small fish community survey in Lake St. Clair. Next slide, please. All right, for the results and discussion portion, you can see a graph that I made to display some of the data trends that I've been seeing when comparing the number of recaptures in both lakes. The data range for these recaptures range from about 2002 to 2019. There was no data from 2020 for obvious reasons, and data from 2021 is still being collected in the field and has yet to be added to the database. Lake Michigan, being a much larger body of water, is statistically going to have higher numbers as displayed here. Interestingly, of almost 4,000 recaptures in Lake Michigan, only about 9.75% were recaptured on more than one occasion, and it was only about 6.2% for Lake St. Clair. As I continue to research this further into the fall semester, I want to take a look at the movements of the smallmouth bass and how far they travel between these recaptures, as well as explore possible reasons for these low recapture rates. Next slide, please. All right, working at both fisheries with the DNR has been a truly wonderful experience and has given me a better idea of what I would like to do for my career after I graduate in the spring. As I have mentioned already, I plan to continue this research to the end of the summer and into my fall semester for my environmental science internship class. I hope I can continue to collaborate with the remarkable team at the DNR and maybe one day join their team full-time. I would like to say a huge thank you to everyone at the Michigan Sea Grant team and my advisors, Dave Clapp, Todd Wills, and everyone else at the Lake St. Clair and Charlevoix DNR fisheries that guided me through this internship and who are always willing to lend a helping hand. Thank you.
Thank you, Cheyenne. That was a great presentation. It was exciting to see you holding a gar, I think, right?
Yeah.
We don't see very many of those. Great. So up next, we have Elizabeth Bellinger, and she's looking at an interesting project looking at aquatic ROV sensors. And she's with Lake Superior State University.
Yep, thank you for having me. And my name is Elizabeth Bellinger. I'm a bio major and a robotics minor. I did my project through Center for Freshwater Research and Education, who gave me tons of help, which is through Lake Superior State University, and my mentors were Dr. Sarda and Dr. Kat. Next slide, please. So I wanted to just jump right into the background and the purpose. And some of you may be wondering, what is an ROV? An ROV stands for Remotely Operated Vehicle, which in this case is an underwater remotely operated vehicle. And you can see in that top picture there, that's the ROV I was working with. That's called the SOFAR Trident. So what is the purpose of my project? So the question was, is there a less invasive way to get population data of lake sturgeon? As the current way, they take population data through set lines, which may have somewhat of an invasive factor. So we are wondering, will the ROV be an effective tool in locating these sturgeon in a non-invasive way, so going to the sturgeon? And if it is, can we add sensors to collect real-time data where their sturgeon was found to get more information on their habitat? What was achieved in the summer was a complete prototype, which includes the housing for the dissolved oxygen sensor and the turbidity sensors, as well as the electrical components within the slide 2. Next slide, please. So I want to go over the overlook of my project. There was 3 main aspects, which is the manufacturing, electrical, and the programming. When it came to the manufacturing, I had to design mounts for this watertight enclosure. So we ended up purchasing a watertight box and modifying it so the sensors and electrical components could be housed inside. And that is what was mounted onto the ROV. And I modeled this through a computer-aided design called Creo Paramedics, which you can see from those top 2 pictures as my model designs. And with the help of LSSU, I got these parts 3D printed. The hardest part of the manufacturing was definitely the buoyancy. This is because that box held a lot of air, so I had to find ways to distribute the weight to offset that, which took a great amount of time, but I did end up getting that. When it comes to the electrical side of my project, we use these low-cost DIY components from a community-based company called Enviro DIY. These parts are really easy to alter and are made specifically for environmental data collection purposes, so they were probably the best ones to use for our purposes. The hardest part of the electrical was getting a good connection because of how small the enclosure was, which you can see there on the bottom picture. There was a lot of wires and a lot of components to put in there, so trying to get the wires to stop poking out was pretty difficult. But because of this, I learned a lot more about how wiring works, how it transfers data, and it was even my first time learning how to solder with the help of C-Free. They helped greatly. When it came to programming, because these were DIY sensors, they had to be told what to do, simple things that we wouldn't think would need to be told. So how many samples to take, at what intervals to take these samples, stuff like that. The hardest part of this was I didn't really know what the program interface was before I started using it. It was called Platform IO through Atom. So it was definitely a learning curve on that. But because there's so many resources through the help of LSSU C-Free and my mentors and Enviro DIY, I was able to put a code together from a bunch of references. Doing this definitely helped me understand the interface and the language. And I learned a lot more about how to work the file directories and other programming components like variables. Next slide, please. So a little bit more in the design process. On the left, you can see my first prototype there. Because there was a lot of shipping delays, I could not receive my electrical components, the box, or the sensors till midway through the project. So the majority of my first project was designing on CAD, 3D printing, going through iterations to find what was best so that when I do get my parts, I could tweak out all the flaws. You can see that I added weights on that 3D printed on the left picture to distribute the weight how I thought it would be distributed for later to test my buoyancy. And then on the right there, that's my current final design. I'm still going to be continuing with it. Every design can be improved, but it's changed in ways, but it definitely, the general design stayed mostly the same. Next slide. Going a little deeper into the design process is all the iterations. As you can see, the box mount went through the most iterations. Pointing you to that second photo there, that red box mount does not have a horizontal flange like that orange box mount. I think that was one of the most notable changes that I did. The sensor mounts, they did not change very much. They got thinner and offset a bit. In that last picture, there's the tail fin. That is still being in consideration if it's beneficial or not. So I'm still running tests to see whether I should add that onto my final piece or not. Next slide. So because this was a super intensive project for the summer, I did not get a chance to go into the biological aspect of my project. However, what I've done in the summer is the base start of my senior research project that I will continue through my years of LSSU. So I'll continue improving the prototype, and then when it's ready to take data from my senior research project, I will attempt to answer the same biological question. Will an ROV be effective in locating lake sturgeon and get intel on their habitat? So my project will continue through about the next 2 years. Next slide. And I have some videos. I hope they work. I'm not sure if they will run, but of some of my test runs with the tail fin and without the tail fin. Oh, yeah. So I want to thank you for listening, and thank you to all the help that I got from CFRE, from the school, from my mentors. There was a lot of resources out there to make this happen. So, thank you.
Nice job, Elizabeth. Great presentation and cool videos that you had to show your work. I know this isn't exactly what you thought it was going to be, but you gained a lot of really great experience in engineering and programming. So good work. All right, up next is Simon Freeman, and he's also with Lake Superior State, and he's looking at the relationship between larval whitefish and zooplankton density in 3 different lakes. Take it away, Simon.
All righty, thank you very much, Catherine, for the introduction. Yeah, my name is Simon Freeman. I'm a fisheries management senior concentrating up here at Lake Superior State University. And I use the Michigan Sea Grant and in partnership with Lake State to look at my undergraduate research, quantifying some day versus night relationships between larval lake whitefish and zooplankton in the upper Great Lakes. Next slide, please. So the lake whitefish, we know, a very commercially, culturally, and recreationally important species in the Great Lakes, but they've been on the decline since the 1990s. And this is largely due to invasive mussels coinciding around that same time arriving in these upper Great Lakes. They decrease phytoplankton populations, which then decrease zooplankton populations. And zooplankton are a critical food source for larval lake whitefish, and decreased food for these larval lake whitefish can limit their recruitment to the next life stages. However, we do know that habitat used by zooplankton and fish can change drastically at night versus day. And so if we are only sampling during the daytime, are we missing something at night? Are our population estimates going to be a little bit different? So we were able to add kind of both of these factors into this project. Next slide, please. So what is the relationship between larval lake whitefish and zooplankton prey density and biomass in Lakes Michigan, Huron, and Superior? And are there day versus night population estimate differences for larval lake whitefish and zooplankton? Next slide, please. So for this project, we hit the beach. I myself and several other collaborators on around the Great Lakes region got in the water as soon as we could after ice off, and sampling continued through early June, kind of the time that these fish are hanging around the beach and feeding on zooplankton. This was paired day-night sampling, so we took net tows for zooplankton, 30-meter horizontal tows. We took the zooplankton back to the lab, identified, counted, and measured them. And then for larval coregoneids, we took 20-meter Newston net tows, counted and measured those in the lab as well. And we'll be sending those coregoneids to the USGS for genetic analysis to look at what type of coregoneids we're seeing. And you can kind of see in these pictures here some action by the Straits of Mackinac on both sides, Lake Michigan and Lake Huron, and then some good night sampling pictures there too. Next slide, please. So this image here gives you a good idea, a little bit of just a taste of the spatial extent, and then the sample size too. So there in the Eastern UP, us here at Lake Superior State covered a lot of those points down through the Straits, the St. Mary's River, and then Lake Superior. In Lake Michigan, we had help with U.S. Fish and Wildlife Service in Green Bay, Wisconsin, providing some data, and also the DNR Research Station in Charlevoix, Michigan, provided data points. We do have many more coordinates to add, many more samples coming in as we're coordinating some retrieval of those samples. And a lot of those are from our tribal partners, and we'll add more to the Lake Michigan side of things. Next slide, please. So some preliminary results for now. We were able to process some of the fish here, and we plotted it out on a log scale. However, the numbers on the axes are the actual fish counts, just to put that into perspective. The farthest point to the upper right was 347 larval coregoneids found in just one 20-meter Newston net tow at night. So we plotted them out, and we saw 13 times were the nighttime counts higher than the day counts. Only 2 times did we see coregoneid counts higher during the day, and 1 time was split right down the middle. And to kind of standardize that even more, we looked at percent difference and found 370% higher coregoneid count at night versus day. Next slide, please. And so for the zooplankton, much of my time actually was spent learning the taxonomy and kind of getting as proficient as possible on this skill. And then we got into very briefly some samples here. So higher at night, just looking at density, we found a 2-to-2 split over 4 samples. But then looking at biomass, you can see, different zooplankton, larger zooplankton migrating possibly to the Great Lakes shorelines in the evenings. And that gave us a different stat there, 3-to-1 for higher at night and then higher at day. Next slide, please. And so the next steps, we have many more samples to process, and I'm going to continue that into the fall as I continue my coursework here at Lake Superior State. Approximately 200 samples total, and that's a combination of, coregonid samples, zooplankton samples for night and day. We'll get genetic information on those coregoneids back. We'll look, are they lake whitefish? They could be Cisco, they could be round whitefish, your Menominee. And we'll perform, several more statistical analyses on the day-night difference. And we'll look at it, lake to lake, Huron, Michigan Superior, and then bring it all together and see if zooplankton are really driving this or not. Next slide, please. And so, yeah, a big thank you to all my collaborators on this project, but also Lake Superior State, the Center for Freshwater Research and Education, my advisor, Dr. Jonathan Dobeck, some sampling help that I had up here in the Eastern UP with Jason Smith, Hallie Grulke, a Lake Superior State student, the Michigan DNR, Jory Jonas down there, also the Little Traverse Bands, Chad LaFavor, Grand Traverse Bands, Chris Hessel, and also U.S. Fish and Wildlife Service over in Green Bay, Wisconsin, Sharon Rayford. So thank you to everyone. It's been a great professional development experience on its own and a very nice data set to work through. And thank you to the Michigan Sea Grant as well for this, this opportunity.
Well, thank you, Simon, for a really nice presentation. It's great to see you have such great collaboration and working on. I know whitefish are a really important issue in the Great Lakes, so I'm glad you're able to contribute to our knowledge base there. Thank you. So next we have about 5 minutes for people to ask questions of Rachel, Michaela, Cheyenne, Elizabeth, and Simon. So you can put questions in the chat and Emily will convey those, or you could raise your hand using the some tools and some Zoom tools that I don't really see in my little set of things right now. But.
Yeah, so we'll probably have 5 minutes, maybe time for 2 to 3 questions. But if your question doesn't get asked, we can answer that at the end Q&A session.
Great.
So I see Geneva, you have a question. You can unmute yourself and ask.
Thanks. I just wanted to let people know if they want to raise their hand, they can click on the reactions button down in their Zoom toolbar and they'll access the raise and lower hand function there. Thank you to all the students. These presentations were awesome and it's great to hear about your work and I'm looking forward to hearing from everyone else. Rachel, I was wondering if you could talk a little bit more about your partnership with the Sioux tribe and what activities you're able to do with the tribal members.
Yeah, so it was a great experience. I was able to go up to Sault Ste. Marie, which I've never been to the UP before, even though I've lived in Michigan basically my entire life. But with Jason Smith up there, I was able to go boomshocking twice throughout the night. So we went out at like 8 p.m., came back at like 2, 3 a.m. That was definitely an experience and it was probably the most fun I've ever had fishing, even though it was so late at night. But yeah, so my coordination with the tribe was essentially just to have an opportunity to be able to sample in their waters. So I was able to fortunately coordinate with their team and yeah, we caught some really cool looking fish. I think I got 5 from that site, so it was awesome.
Okay, then Rachel, we have another question for you in the chat from Victor. He asked, are those viruses native or endemic?
I am not sure if the largemouth bass virus is a virus that is well known in Michigan to cause disease and mortality events in largemouth bass. Now we're seeing more with smallmouth bass as well, but with Flabobacterium columnare, the same thing. These viruses are well known in Michigan. I well known virus and bacteria in the Great Lakes.
If anyone else has any questions, you can ask in the chat or raise your hand. And if not, we can move on to the next section of presenters. Okay, Tom, I see your hands raised. You can unmute yourself.
Yes, thanks, Emily. So I have a question for Simon. So I wonder, based on your work, do you think it's advantageous for folks that are trying to assess how plentiful larval Coregonids are to maybe start sampling more in the evenings or nights, as opposed to, like, are our estimates off? I mean, I think I understand that was the gist of one of the gists of your research, but do you have any thoughts on that?
Yes, thank you very much for the question. Very good question too. And one that I've given a lot of thought on. You know, I look at many sites and, you know, we see that percent difference, 370% higher. And that's just in, you know, this 15, 16 fish samples we processed thus far. And we have these data sets coming in and talking to collaborators, and they're saying just visually, we're seeing more Coregonids at night. And, you know, that could be a predation technique for these fish to be on the beach in the evenings, you know, but, you know, where are they going during the day then? If we're going to, you know, look at lake modeling and seeing how many, you know, how strong year classes are year to year, if we go out and miss them during the day, but we go back to that spot at night and see hundreds, then maybe that year class isn't as bad as we think in certain areas. A specific spot, the Straits of Mackinac were a spot of, you know, they're an area of high concern, especially in the zooplankton community, but Coregonid wise, we've seen a drop off in that fishery in that popular area. But we saw, you know, very few fish during the day and then hundreds at night on the Lake Huron, side of the straits. So, and that's right up against the bridge there. Like I said, right through that area of concern. So our estimates off if we just look at those day numbers, especially from that spot, but others as well. Thank you.
Okay, we have time for one more question, and this question's for Michaela. So Michaela, did you come up with the idea of dog food as bait, or is that a common sampling strategy?
So I did not come up with that idea. It is a common sampling strategy, especially gobies. They'll eat just about anything. Another bait we had used was whitefish, and just had those in the minnow traps. We did see those probably got a higher capture rate for gobies, but yeah, they'll eat just about anything.
Okay, great. So that's it for this Q&A session. Like I said, if you have any more questions, you can ask at the final session. But right now, we're going to move on to our next group of students.
Thank you, Emily. So coming up next is a group of students that, you know, in general, they did field surveys, they looked at creating information and environmental education programs. And we'll start off with Ethan Valkenburg. He's with the University of Michigan with advisor Ben Winger. He's looking at conservation of Great Lakes coastal wetland bird communities. Take it away.
Hello.
Hi.
I'll be sharing some of the work oops, sorry. Okay. I'll be sharing some of the work I did this summer on Great Lakes coastal wetland bird communities. Before I begin, I just want to thank my advisor, Ben Winger, for meeting with me throughout this project, the Biostation, the University of Michigan Biostation, for giving me a place to do this work, the Great Lakes Coastal Wetland Monitoring Program for sharing some of their data with me, as well as the Michigan Sea Grant for your support. All right, next slide, please. So Great Lakes coastal wetlands are threatened by a lot of different things, including intensive agriculture throughout the watershed, urban runoff, invasive species, fluctuating lake levels, and a changing climate. And this is putting a lot of pressure on the health of these ecosystems and on a lot of the native species, including birds that rely on the coastline during breeding and migration. A recent study funded by the Audubon Society identified the area near the Straits of Mackinac as an area of extremely high priority to protect these wetland birds. So the purpose of my project is to answer this question of what factors influence those marsh bird communities and how can that inform our strategies for conservation and those priorities as well. Next slide, please. So to answer those, back one. To answer those questions, I lived at the Biostation and surveyed wetlands identified by the Great Lakes Coastal Wetland Monitoring Program. I used their marsh bird survey protocol at each of those sites and then estimated major vegetation and land cover within 100 meters, conducted invasive species transects at each of those wetlands, and then collected landscape variables from the National Land Cover Data Set at a scale of 2 kilometers around each point count. And once I sandwiched my data together with that of the monitoring program, I ended up with over 487 points and over 11,000 bird observations. So quite a large data set that I was excited to work with. On to that next slide, please. So to start and answer some of these questions, I subset that data set to include 14 focal marsh obligate species and used multiple linear regression with those candidate variables listed below to predict the marsh bird abundance at each point count. You can see there with the bullets, those indicate the direction of the relationship, and the asterisks show the significance of that predictor variable. And I've plotted those 4 most significant predictors on the right there. So just note that wetland size, the proportion of cattail within 100 meters, the proportion of emergent wetland within 100 meters were all positively related to marsh bird abundance, whereas human development within 2 kilometers was negatively related. Onto that next slide, please. So I was curious whether all of these species were responding similarly to these different variables. So I subset the data even further to include marsh obligate species with sufficient detection, and then used a random forest classification tree method to predict the occupancy of each of these species. And here I've plotted the importance of each of those variables. So in the interest of time, I'll just draw your attention to how different the important variables are for each species, and then how something like wetland size is very important for nearly all of those wetland bird species, and something like the proportion of grasses is not very important for any because it doesn't provide the structure and cover that something like the proportion of cattail cover might. I'll also draw your attention to that value or the variable year, which is a bit of a stand-in for lake levels because those can change annually and have changed significantly over the past 10 years. And that seems to only affect a lot of those small wading bird species that live kind of at the margin and nest at the margin of kind of these wetlands and the open water. Onto that next slide, please. I also, moving forward, I want to look into how all of these variables might interact with each other. And one thing that I noted recently was how important this interaction term is in multiple linear regression between this variable of the proportion of open water and the proportion of vegetation. And that I took to kind of indicate this idea of interspersion, which describes kind of the boundary between the wetlands and the water. A wetland with a lot of interspersion would have a lot of pools and kind of a very complex boundary. And that offers great habitat for birds, for their nesting, as well as their feeding. But it varies so much and it's difficult to measure. So that's one variable that I'd like to better include. And you can see on that plot on the left, as the proportion of open water at a point count approaches 50%, the slope of the line that shows marsh bird abundance increases as well. So that's one thing I'd like to explore and maybe develop a more robust variable to represent interspersion. Next slide, please. And just to finish it off, some of the conservation implications from my work, I think really indicate the importance of large wetlands, and not just large wetlands, but maintaining and rebuilding the continuity of wetlands. My wetland size variable was the amount of wetland cover within 2 kilometers. So those adjacent wetlands are just as important. We should also consider buffer zones around wetlands to mitigate the human impacts that occur outside of them, but still kind of trickle down and can impact wetland bird communities. And then we need to be careful in designing how we control invasive species, because this invasive cattail, Typha X glauca, forms these really dense monotypic stands of cattails, which do provide great habitat for some birds. So it would be irresponsible to just remove them all. So we need to develop management strategies that kind of promote interspersion and more diversity of vegetation. And then I think my research also kind of notes the importance of thinking about conservation locally as well as regionally for marsh birds, and also indicates the different, the importance of different variables for all the different bird species. Once again, thank you to the Michigan Sea Grant.
Thank you, Ethan. Great presentation and really interesting work. That's a lot of wetlands to have visited over the summer.
It was a fun adventure.
Yeah, a lot of great field work there.
Oh, yeah.
Okay, coming up next here is, this is one of our sponsored projects by Herpetological Resource and Management Group. This is Giovanni Rosselli. He's at Michigan State, right? And he did assessment of the herpetological habitat assessment tool. Take it away, Giovanni.
Thank you. Once again, my name is Giovanni Rosselli. I work with Herpetological Resource and Management. For my project, I worked on field testing the Herpetological Habitat Assessment Tool, or HERPAT for short. This tool was designed to be utilized by regulators, managers, natural resource professionals, both the public and private sector, and land stewards. And the tool is designed to evaluate the functional value of a wetland with an emphasis on herpetofauna in Michigan. The HERPAT could be a critical tool for helping to protect and best manage landscapes for herpetofauna in Michigan. I surveyed 13 wetlands along a linear project area, utilizing MIRM tests, herpetofauna observation data, aerial imagery, and GIS desktop analysis. The herpetofauna data included species richness, abundance, and species-specific coefficients of conservatism scores, or C-scores. We compared MIRAM, HERPAT, and combined scores for each site and analyzed the data based upon correlation and linear regression. Next slide, please. This image over here is an example of one of the test sites that I surveyed. The box indicates, the boxes were used to locate wetlands along the corridor that had a significant enough data set, a large enough number of observations to run these tests. And we found that a negative correlation of negative 0.3579 between MIRAM and HERPAT scores. A linear regression showed an R-squared value of 0.1281, a negative 0.017 coefficient, and a p-value of 0.2298. We thought that a small sample size could account for the small R-squared value. We only had 13 samples. A previous desktop-only review conducted by HRM had an R-squared value of 0.094, a negative 0.17 coefficient, and a p-value of 0.05, which would be statistically significant. And due to the desktop-only review supporting a statistically significant relation between MIRAM and HERPAT scoring methods, a desktop review with a robust enough data set could be an important tool for justifying future field sampling at a site. Next slide, please. Amphibians and reptiles engage in resource partitioning, are at a medium trophic level, and are sensitive to changes in environmental conditions. Due to this, they're both useful indicators of wetland functional value. The results of this survey suggest that the detection of herpetofauna is higher at sites with lower MIRAM scores. This is in reference to the negative correlation that we found between the two scoring methods. And wetlands with a history of disturbance can still have high functional value for herpetofauna. That was one of the key driving factors behind developing this tool, was that MIRAM tends to favor large, contiguous, undisturbed habitats to gauge the functional value of a wetland, when oftentimes we find diverse communities of herpetofauna at much more disturbed or degraded habitats. These wetlands can still support herpetofauna diversity, and this tool aims to showcase that. For future improvements, developing a scoring category that accounts for multiple age classes of a species at a site to show that a given site, say, it supports larval forms of a frog as well as juveniles and adults, would score higher than a site that only supported the larval form. Larger sampling sizes for more normalized data would always be helpful. Surveying entire wetland complexes instead of small representative areas would be helpful. Developing a modifier to account for sites or species with low detectability, and testing the correlation between individual MIRAM metrics and HERPAT scores to see which submetrics of MIRAM might affect HERPAT more than others, I suppose. I just realized I never mentioned MIRAM is Michigan Rapid Assessment Method. It's a tool developed by Eagle to gauge the functional value of wetlands. And I'd like to thank Michigan Sea Grant and HRM and David Mifsud, my mentor, for this incredible opportunity. Thank you guys very much.
Thank you very much, Giovanni. That was really, really interesting. Look forward to hearing more about it. Good presentation as well. Next up, we have two presentations that are the third one of our sponsored projects. And this is a project led by Mike Reed from the Detroit Zoo. And the person who was talking about one part of the project here is Gabrielle Dumont. She's from the University of Michigan, talking about environmental education projects that she was a part of and helped develop in communities in Detroit. Take it away, Gabrielle.
Hello. Like Catherine said, my name is Gabrielle Demott, and this summer I was able to work with the Detroit Zoological Society to engage communities in Detroit with environmental stewardship and just to help people learn about the environment and get outside and connect with their surroundings. So I was fortunate enough to work with Mike Reed, who's pictured left, and we focused on a couple specific aspects of community engagement. Catherine, could you go to the next slide, please? Thank you. So one of the main things that I did this summer was to create lesson plans that can be passed on to different agents in the community and implemented in various ways. Some of this might include online activities for families or programs done through community centers. We were able to test out some of these activities with the Green Door Initiative, which is a group that I'll talk about a little bit more later on. And then a big part of the project was doing frog watch training and creating an educational video for other citizens who want to engage in science. Next slide, please. And just to highlight a few examples of the lesson plans that I created, one of them had to do with creating a plan for cleaning up trash in communities that people without a science background would understand and relate to. So part of this was explaining why trash is bad and how it can affect things that people can see and understand. On the right are just a couple slides from a presentation I made that can be presented to a variety of community groups, detailing some of the specifics of waste management and trash cleanup in Detroit specifically. And then I was able to actually implement and practice this with some members of the community at a beach cleanup at Belle Isle, where we focused on one of the lakes there and picked up trash around it and explained to members of the community, some of which were children, which posed a different challenge, why it's important to do what we were doing. And then we categorized all of the trash we found. So we have that information for future purposes. And next slide. Another lesson plan that I created has to do with the Detroit River's unique position as an international waterway. So I put together a lot of information about countries that ship through ports in Detroit that use the Detroit River and how to identify their flags, what imports and exports they do. You can see that on the right. There's an example there. That way, people who are out in the parks are able to learn a little bit about the Detroit economy and the function that the Detroit River serves beyond beyond an environmental sense, also in a historical and a geographic sense. But then I also included a lot of information about invasive species and how they get transported in through waterways so that people are aware of how that happens and what they can look out for when they're in the parks and how it might be damaging the spaces that they're enjoying. The picture on the left is from A.B. Ford Park, which is directly on the Detroit River. Next slide. A big ongoing project that we did throughout the summer was Frog Watch, which is a citizen science program that enables members of the community to monitor frog populations in their own area. So the first thing that we did was to get certified. It was a couple hour long online training program. And then we went to Belle Isle one night and did a practice observation so that Aiden and I could get familiar with how the Frog Watch observations happen. And then we were able to make a video that breaks down the process in a very accessible way for future efforts in getting the community involved in this sort of project. So that's us on the bottom left filming this video. And then on the right is just a frog that I saw when I was at Belle Isle one day, and I took a picture of it. Next slide. A big thing that we did towards the end of the summer was working with the Green Door Initiative, which is a program in Detroit that focuses on getting kids in the city to go out and interact with nature, to build a passion for the environment, to engage them with stewardship. And so we did a couple different things. On the bottom, we did a nature hike where we took them around A.B. Ford Park and worked on identifying species with guides that Aiden will talk more about. The top left is a nature hike that we did on Belle Isle, where Mike led us through this part of the park that I think a lot of us had never been through, and we got to learn about some of the ecology of the area and also how to engage people with that sort of situation. And then in the top right is a macroinvertebrate monitoring that Aiden will also talk more about. But we were able to get the kids involved in digging through the mud, basically, which doesn't sound super appealing, but they really liked it. And it was very rewarding to be able to connect them with their surroundings and explain to them why things like this are important for their health and for the health of the ecosystem. And next slide. Okay, that was the last slide. But I am really, really grateful that I was able to work with both the Green Door Initiative and the Detroit Zoological Society and all the people at the Belle Isle Nature Center as well. It was incredible just to be able to go to Detroit a couple times a week and meet with different community groups and sort of spend some time experiencing like nature and the environment from the perspective of people that aren't scientists or who don't have a science background, and just getting to experience their passion and their excitement for the things that we were doing. It was really, really rewarding, and it's something that I want to continue to do in my career. So thank you very much to Mike and to the DZS for that opportunity.
Thank you so much, Gabby. That was a really nice presentation, and I'm glad you were able to get kids excited about digging in mud for aquatic invertebrates, since that's what I love to do too. All right, next up, we have Aiden Baubeck, and he's a student at Michigan State University that also worked with Mike on these environmental education projects. Take it away, Aiden.
Hi everyone, I'm Aiden Baubeck. So like we've said, I worked with Gabby and under the supervision of Mike to develop some environmental lesson plans and activities to help engage the east side of Detroit community in really just getting out into nature and appreciating it. So yeah, inspiring environmental stewardship. Next slide, please. So one of the major things we did was we worked with Frog Watch USA, which, like Gabby said, is a citizen science initiative run through the Association of Zoos and Aquariums. So we were trained to identify the 14 frog species of Michigan by their calls. And we are working to put together a training video to help encourage people to go through the training, but also to help give a visual for the training of how to conduct the process properly. Next slide, please. So one of the major places we worked was A.B. Ford Park, which is a little park on the east side of Detroit, right along the river. And they're currently in the process of building a new community center there. So a few of the various lesson plans that we wrote can be used there. One of them was the scavenger hunt that I put together. And the point of the scavenger hunt is to help people of the community, and it can be anyone, kids, teens, adults, they can go around and learn to identify some of the different plants, and especially the edible ones. So like mulberries, or like what plants to avoid, like stinging nettle or poison ivy. Yeah, and so all these different activities are going to be very useful to help just encourage people to learn more and give them a starting point of being able to identify these species. Next slide, please. So my big project for the summer was kind of working on a guide for identifying macroinvertebrates. And it was written in a way that's very accessible to anyone, regardless of experience with science or macroinvertebrates. You know, usually guides like that tend to be a little more on the colder side, and it can be kind of hard for just the common person to understand. But it was written to try to be relatable, and anybody can pick it up and learn more about macroinvertebrates and be excited to go out and look for them. So both the macroinvertebrates lesson plan and the scavenger hunt, we worked with the Green Door Initiative, and they both seem to really like both of the lesson plans. Next slide, please. So here it is. The cover page is kind of just defining what a macroinvertebrate is, why you should care. So a lot of these can be used as indicator species for ecosystem health. So there are 3 different levels. The first level is very pollution sensitive, so you need super clean water to live in. Next level is intermediate, and then the 3rd level can usually live in dirtier water. So teaching people about these allows them to go out, look for these macroinvertebrates. And if there's a whole bunch of these species that require the super clean water, that can help show that, you know, their ecosystem is healthy, and allow people to kind of check that themselves. Next slide, please. And that's all. I'd like to thank this Michigan Sea Grant. I would like to thank you all for coming to the symposium, coming to hear us all present our research. And I'd like to thank Mike Reed. I'd like to thank the Detroit Zoo for allowing us to do this internship this summer. And I'd like to thank Belle Isle for allowing us to use their nature center and do some of our programming over there.
Great, thank you, Aiden. Great presentation, and again, exciting to hear about the macroinvertebrate survey that you put together. And also, I thought the idea for having a scavenger hunt to teach people about plants was a great idea. So nice presentation. Okay, up next is switching gears just a little bit here. We have a presentation by Angela Gutierrez Vargas. She's at Michigan Tech University and looking at socio-ecological stewardship of a tribal landscape. Angela.
Hi, my name is. Hey, thank you for the intro. My advisors were Evelyn, Dr. Baird, Dr. Val, and Marissa. I would also like to give like a quick shout out to Kathy, Joe, Zena, Karina, Jerry, Katie, Carlos, Mayra, Stephanie, and Kelly Raffaelli for all their guidance and mentorship throughout my time here at Tech. Without them, I wouldn't have made it this far. So before I begin, I'd like to kind of give a little background of the kind of presentation I'll be giving. It's guided by the principles of Photo Voice. Those unfamiliar with Photo Voice, it's a community-driven method of communicating ideas, and it allows participants to engage in active dialogue and critical reflection, and then create community change. And in order to stay true to that Photo Voice principle, one of the big things to do is acknowledge your position as a researcher within the community that you're working with. So I'm a Chicana Latin scholar. I am not a tribal member. And I think this is important for me to start off with because it opposes historical and abusive extractive research methods that have harmed many BIPOC communities, including my own. Next slide. Thank you. So this summer, I wanted to continue my organizing work and also make connections with the KPIC, NRD, the Natural Resources Department. I used their research guide to basically align their goals with my research. I'm a guest in their community, and they know what is best for their community. So I wanted to prioritize their goals and vision and align my research with their needs and vision. I would also like to acknowledge all the voices and scholars that guided me through the summer. They're on the left here. They're just some of the works that I've read. Their works have paved the way for future generations of scholars like myself. This summer, my research is in 4 parts, is under the guiding principle of environmental justice is racial justice. I continued the BIPOC garden space. I went to the Treaty of People gathering. I did the signage, and a little bit of work with the National Resources Department. I also did my own personal artwork. I'd also like to define or give a little bit of background for those unfamiliar with environmental justice. Environmental justice is essentially a concept that groups various ideas of equity over the distribution of power over the resources. And I think power here is key. It allows us to think about who controls the resources, who decides how to use them, who is excluded from these decisions, and who suffers the consequences of these decisions. And this is not just oriented to people. I argue that it is our ethical obligation to equitably distribute the power to more than human beings. This is not a new thing. Indigenous people have been thinking about these ethics since time immemorial. Next slide. The BIPOC Garden it's years in the making, really. I wanted to build off of my previous organizing. It was the pandemic, and I was reading some statistics about food insecurity in the BIPOC community. BIPOC means Black, Indigenous people of color people. And there was statistics about food insecurity, and we suffer the most food insecurity of all students during campus. Then I read a 2018 survey done by Tech that said that 30 to 35% of students suffered in food insecurity on my campus. They didn't include any racial demographics, like unsurprisingly, but statistically speaking, I knew that the majority of people, the students, were BIPOC, statistically speaking. And so we wanted to create a space to grow and share community, and we did the garden. Next slide. The next part of my summer is I went to northern Minnesota, and I was a guest in the White Earth Reservation. I got to engage and witness a real-time emergent protest zone. I learned about nonviolent direct action, and I participated as traffic control, and I decongested like critical areas of movement. I learned about the history of treaties, colonization. I gained a better understanding of extractive industries, and I got to witness a protest site become essentially like a collective movement for change. And it was really powerful. Next slide. The third part to my summer is that I got to work with the Keweenaw Bay Indian Community Natural Resources Department. The staff there was wonderful. I got to join for a day with the youth program and do a little bit of restoration for stamp sands. And I also made a huge poster of the slides that some of the high schoolers made. And then during this time, I also gathered tons of information and context for the next part of my research. Next. The final part of my summer is that I worked on making signs for Sandpoint, just within my own artwork. And I'd like to mention that my art comes from a very deep sense of vulnerability, since I was raised with Marianismo. So it basically means it's a really racial and extremely gendered issue that has roots in colonization, because it forces women to just push many topics of pain, loneliness, isolation, loss, and dehumanization for the sake of keeping peace. And for the KPIC, my kind of artwork was telling traditional stories and teachings. And this kind of storytelling is a means of remembering culture and wisdom. This kind of storytelling is a direct action against erasure, against colonization that essentially wanted to murder Indigenous people. And it's a breaking of silence. It's a way to talk back against colonization. It's a way to go back home. My art is how I go home. It's how I talk back. It's my continuous and daily struggle as an act of resistance. And talking back as a Latina means that I'm pushing boundaries, borders, and making bridges as I go. Haciendo puentes al andar. Thank you.
Thank you, Angela. That was a very wonderful presentation, and it sounds like a very personal story that you're sharing. So thank you. And beautiful artwork, by the way.
Thank you. It was an honor working with the KPIC and allowing me to do this kind of story work for them was powerful.
Okay, now we have about, again, a 5 minute time for questions. And after the questions, we'll have a 5-minute break. We'll have about five minutes to ask questions of Ethan, Giovanni, Gabby, Aiden, and Angela. Again, you can use the chat or raise your hand.
So we have a question for Gabby, and this is from Geneva. Geneva asks, what were some of the most common types of trash you found in the cleanup?
Yeah, that's a good question. The specific cleanup that we did was close to a park area, so a lot of the trash that we found was food wrappers, things that people kind of ate and then just discarded. We focused on looking for small plastic things because that was one of the important things we told the kids, was that those small plastic things are really, really harmful when they end up in the water. And that was a way to sort of get them to understand why it was bad and why what they were doing was important, because they were able to see the body of water that we were next to and some of the living things in it, and realize that what they were doing made a difference, which was a really big part of the trash cleanup.
Okay, we have, oh, I just lost the question, but we have a question for Ethan. Ethan, did you find any wetland birds that are uncommon in Michigan?
There were a few that were kind of mentioned as focal species in the protocol, like a night heron that I never did get to see. But I did see a least bittern, and that's one of those birds that isn't super common. But generally, a lot of these marsh birds, a lot of people call them secretive marsh birds because they kind of hide out in the marshes, and they're birds that I had never really seen before spending time in these marshes and like spending hours just kind of sitting, waiting, watching and listening. So to me, there were a lot of new birds and new things that I got to see just by spending time in that space.
Thanks, Ethan. And now the next question is for Angela. So Angela, will the garden be continuous after you leave Michigan Tech, and have steps been taken for others to take over?
So that's kind of like a really loaded question with the environment that Tech has really kind of made for Black, Indigenous, people of color students. Right now, I think Latino students are a little less than 1%. I believe Tech, it's similar demographics for Black, and I think Native Indigenous students make up like less than 0.001% or something crazy like that. So really, this kind of work is headed by just community organizing leaders, and whether I would love for it to continue, but just by the mere population that Tech has, probably not. But hopefully it helped people. It helped the people that it got to reach. So yeah.
Thank you, Angela, for that question. Next one is for Ethan. Your graphs showing the importance values of marshland birds don't add up to 100%. How is the importance index weighted related to the other variables being considered? Examples, years, size.
Yeah, I think I accidentally put an older version because I was having some issues with those charts at one point, and I may have forgotten to switch those. But importance is, it's hard to explain, but there's this thing called the impurity of the data set, and after each split is decreasing the impurity. So importance is a measure of the average decrease of impurity for each variable across all of the trees that are constructed for each forest of regression or of classification trees. And they should add up to 100. Again, I think I accidentally put a previous version where the scale was a little off.
Okay, so for our next questions for Gabby and Aiden. Can you both talk about the current relationships between Belle Isle and the Detroit Zoo? Historically, there was an aquarium, but not sure about now.
So the way that it works is that they're kind of tied in together. So the Detroit Zoo is the main institution, and Belle Isle, specifically the Nature Center at Belle Isle, is kind of run by the Detroit Zoological Society.
I believe the aquarium is open now. The last time we were there, it seemed like it was back open to the public, as well as the conservancy with the gardens. So if you are in the area, definitely check it out. It's beautiful.
Okay, then we only have time for one more question. So this question is going to go to Angela. So Angela, could you please elaborate on how your crucial ethical practice might help Latinx and other researchers and collaborators work as guests in Indigenous communities? Specifically, they're focusing on how this can help us accumulate crucial data and interpretive details.
Yeah, I'd first like to answer that question by saying that it's not, I didn't make this ethical practice. It was shared to me by one of my advisors, Larissa, and it actually has origins in China, about the photo voice method, but just ethical practice as in like knowing and being aware of your position, I think, and then might help Latinx and other researchers. Well, I think just knowing about that power structure, the power dynamic, and not going into communities and saying like, oh, you guys need help, I see that you need this and that. No, like actually letting them have agency over what goes on in their community, and you helping them work alongside with them. And I see this a lot in, like, I'm from Detroit, so I see a lot of gentrifiers coming in and taking all the grants and then just leaving the city. And I hope that answered your question. Just work alongside, don't be a white savior, essentially. You know, don't be a savior. That's just my personal reflection on this question. I hope that answered your question.
Okay, thank
you so much. You know what? And I think that since we're a little bit behind schedule, we have only about half an hour more. I think we can just skip the break and go on. Hopefully that's okay with everybody. I just want to make sure that we get time in for these last three presentations and time for the final questions and answers, because there's been some really great questions. And so I hope that's okay with you all, just because we're a little bit behind. So coming up, moving right on here, coming up next, we have the last three presentations are really focused on green infrastructure and contamination issues. And we'll start off with Grace Mapes. She's a student at Wayne State University, and her advisor is Daryl Pearson. Take it away, Grace. Is Grace here?
Hi, everybody.
Okay.
Thank you. Throughout this summer, I worked with Wayne State University's Facilities Planning and Management Department to map and document various aspects of the university's trees. And leading this project was the Office of Campus Sustainability. And they helped with, among other things, to provide a platform for my research findings to reach the public, and also facilitate the connection with the Planning and Space Management Department, who provided data mapping support. So I'd like to thank you all for your support, and of course, Michigan Sea Grant and NOAA, who made this project possible. I've also been able to connect with the Ground Services Department to inform various needs for tree care along the way. So all of these connections ultimately resulted in a broad-reaching dialogue on the value of campus trees. Next slide, please. So Now to dive a little bit deeper into the methods of this project. So it started out with the fieldwork portion, which consisted of using ArcGIS Collector app to map and collect the data on a portion of the campus trees. And you'll be able to see the survey area in a map coming up next. So after that, I recorded the service values of the trees using the National Tree Benefit Calculator, and the information that I plugged into there included the species, the diameter at breast height, so essentially the size, and also the land use type and the area code. So it was based on that specific location, the campus location. From there, I used ArcGIS Desktop to visualize the data and the amount of stormwater each tree intercepts on an annual basis, among other things. And lastly, we are working to create an educational program to share these results. My work with the Sustainability Office is continuing into the fall. So it'll be a really beneficial thing to be able to continue the education around the findings into the future. Next slide, please. So to talk a little bit more about the service values and essentially the benefits that we found. So I was able to collect and map and document 492 campus trees. And from there, I recorded each of their service values in the 3 categories, as you can see here on the slides, were stormwater, energy conservation, and reductions in CO2. So starting with the stormwater, we can see that the economic impact is estimated at over $4,000 annually. And I wanted to give a disclaimer to mention that impervious surfaces are taxed in the city of Detroit. So that means that by removing a portion of concrete or impervious surface and replacing it with a pervious area with a tree planted in that place, we'll see additional savings that come from that annually. And we actually see a lot of that type of structure on campus where there's in the middle of a concrete area, we have a tree planted in an area for that space. So not going to read each of these for time's sake, but you're able to see them on the slide here. And I wanted to touch on the results seen regarding the energy conservation. These annual estimates come from previous research that has found that trees on the east and west sides of buildings keep buildings cooler in the summer. And in the winter, the trees create a barrier from weather to reduce loss of heat. So then now looking at the CO2, we see an annual reduction of over 150,000 gallons annually. Now, part of the education for this will be to put these numbers into context for the public. It's really difficult to kind of quantify those numbers if you don't have a comparison to look at, such as for the carbon reduction, such as miles driven in a car annually, something like that. Next slide, please. So the data visualization part of this, looking at this map, you can see a yellow transparent block or polygon. That represents the surveyed area thus far of the campus. And then the solid yellow shapes are the campus buildings. So that's just for context. And we can look at the points that each represent the location of a tree, but also help to show where the most stormwater is being intercepted by these campus trees. So you can see the larger dots, you can see where the areas are that essentially it gives a visual of the larger trees, but also with the larger trees and the type of species, it results in a larger amount of stormwater intercepted. Again, the data visualization will be important in future education efforts. So we can go to the next slide now. Thank you. And to wrap everything up, I'd like to really highlight the importance of finding the connections between nature and the built environment. This research really worked to inform those local connections specifically on Wayne State's campus. So it'll be really helpful in the future to be able to hone in on the local aspect of the benefits from the trees. Thank you all for listening.
Thank you so much, Grace. That was really a great presentation and really interesting work that you're doing. All right. Next up is Cynthia Gutierrez Navarro. She's with Wayne State also, and she's looking at the characterization of ecological processes that stabilize pathogenic microbial communities in green stormwater infrastructure. Take it away, Cynthia.
Thank you for the introduction. Hello, everyone. My name is Cynthia Gutierrez Navarro. I am a biology student at the University of Detroit Mercy. I'm actually the first UDM student to be awarded this Michigan Sea Grant internship, and so it's very exciting, but I'm hoping that there's many more students after me. I want to thank my partners, of course, the University of Detroit Mercy, Wayne State University, Healthy Urban Waters Field Station, Lake St. Clair Metro Park, and of course, the Michigan Sea Grant for making this possible. My project, titled The Characterization of Ecological Processes that Stabilize Pathogenic Microbial Communities in Greenstorm Infrastructure, expands on preliminary findings from a UDM general biology lab in the fall growing season, led by my advisor, Dr. Victor Carmona Galindo. Next slide, please. So for a little bit of background, Lake St. Clair Metro Park underwent a multi-million-dollar Greenstorm infrastructure project in 2013, which is pictured above to your left. This project created a network of 8 bioswales that redirect stormwater into Point Rosa Marsh. It is important to point out that bioswales are anthropogenic and are meant to mimic natural systems. We analyzed E. coli and coliforms across all 8 bioswale sectors and found that all 8 sectors are scoring above EPA limits for recreational water for both E. coli and coliforms. The only significant difference in E. coli was seasonal change. In the summer, we see greater levels of E. coli in sectors 1, 4, and 8, while in the fall or end of the growing season, the greatest E. coli levels are found in sectors 1 and 8. Next slide. Bioswale networks have been overtaken by invasive species, especially frogbit and Phragmites. We know from the primary literature that invasive species can alter bacterial communities. So we quantify bacterial communities with respect to presence absence of both invasive species, one being frogbit that Gabrielle mentioned earlier. And so total coliform levels with respect to frogbit present absence were too high to be quantified. E. coli, however, scored greater than EPA limits for recreational water throughout the bioswale network and did not differ significantly with presence or absence of the frogbit invasive species. No, seasonal change was the only significant difference, which indicates that higher E. coli levels which indicate higher E. coli levels in the summer growing season. Next slide. We also looked at bacterial communities with respect to Phragmites, which is the tall green plant that surrounds me. My advisor likes to say that taking samples of frogbit and Phragmites was like our yoga session for the week, and you can see him laughing. Again, total coliforms in the presence or absence of Phragmites were too high to be quantified, and total E. coli levels scored greater than EPA limits for recreational water across all bioswales. These findings suggest that bioswale networks are not effective at sustaining FIB levels in the summer nor the fall growing seasons. Next slide. So we also took into account abiotic factors such as water chemistry, light, and canopy openness. For the sake of time, I only included one factor here. One of the most interesting things that we found is included in the figure to your left, which shows the relationship between E. coli and pH levels in the bioswales. These findings suggest the bacterial communities may be sensitive to basic pH levels and merit further studies. I hope to continue working on this with Dr. Carmona and throughout the fall season. Thank you for your time.
Great, thanks, Cynthia. That was a really great presentation as well, and exciting to see this really innovative approach to thinking about contamination issues in bioswales. And I apologize for getting your college wrong. We're very honored to support a student from Detroit University. All right, I think this might be our last presentation is from Tristan Compton. He's with Lake Superior State College. I think I got that one right because it's right down there. And he's looking at microplastic presence and density in relation to season land use and storms in the Michigan River network. All right, Tristan.
As she said, I'm Tristan Compton. I'm studying conservation biology at Lake State. This summer, I had a wonderful opportunity to research, collect, and analyze microplastic presence and density in relation to the season land use and storms in the Michigan River system. Next slide, please. There are many different types of microplastics that exist, which can vary in size, shape, and polymer type. In general, microplastics are degraded pieces from consumer items that are smaller than 5 millimeters and come from plastic bags, containers, and even clothes or beads. Next slide, please. Overall, in this project, we looked for the relation between microplastic density, type, and size in relation to land use, season, and storms within the Michigan River network. The reason this question is important in this project is that microplastic density has only been primarily researched in marine environment rather than freshwater systems. Next slide, please. So our methods, we chose 5 sites ranging from the outflow of Lake Superior near Ashman Bay area, to the inflow to Lake Huron down near our detour site at the very bottom there. The three sites found between the outflow and inflow were chosen on their location to either major agriculture, forested, and population areas. These can be seen on the map on the left. Each site was sampled 4 times with a 150 micron mesh plankton net, and each time 2 samples were collected on that site. The samples that have been collected will go through a sieve that is split into 4 separate size classes. These sieve samples then go into a peroxide oxidation to kill off any organic matter that was collected with that sample. Once the samples have been gone through the peroxide oxidation, they are dried and run under a dissecting microscope for the identification of microplastics. Next slide, please. The chart located on the left is put together with preliminary data that helps to indicate the total number of microplastics found from Ashman to Detour. Overall, the microplastics were found on each site, and there seems to be some indication of an increase in our outflow site before the Sulaks, and a slow subtle increase in microplastics after the Sulaks from Osborne to Muscogee. Now in our last site, Detour there, the data was very low because we ended up putting that through the dissecting microscope without the peroxide oxidation. But the sample was saved, and it is going to go through the solution very soon to see if there was any more microplastics. Next slide, please. The 2 graphs below indicate the amount of microplastics found overall and the different sieve sizes microplastics were found in. Thus far in these graphs, we have found that fiber and films were the majority of the overall percent of microplastics, while the percent of microplastics, depending on size, were majority found in both 1,000 to 500 microns. Next slide, please. In the coming months, I will be processing the rest of the samples, at which I will be testing their relation to season, land use, and storms. Overall, about 4% of the research has been conducted on freshwater systems, indicating the need for more research as needed to test the drivers for microplastics in river systems and their resulting impacts on the ecosystem's functionality. Next slide, please. I would like to say thank you to Michigan Sea Grant and the time they have given me in this amazing opportunity. I would also like to thank Dr. Dobek for his assistance and time in collecting and the collection and research of my project. I would also like to thank two SLSU students, Sean Donovan and Cody Heiser, and their help in collecting and processing my first set of samples. And finally, I would like to thank the Center for Freshwater Research and Education in allowing me to use their vehicles and boats to collect my samples.
Great, thank you very much, Tristan. It was a great presentation and really important work to be done on microplastics in the Great Lakes. That's really wonderful. So next up, we're starting to run out of time here, but we wanted to have some time to ask questions for Grace, Cynthia, and Tristan.
Yeah, we'll start with 5 minutes for just these 3 students, and then we'll go to all the students after that. So we have a question for Grace. So Grace, what role does tree species play in your permeability data? For example, the root systems of R selected and K selected trees differ in terms of space and time. That would affect soil porosity, right?
Yes, you are correct. And so basically in the National Tree Benefit Calculator, they've used previous studies that have identified these aspects of the tree water interception. So I actually wasn't looking specifically at permeability data. I mentioned that in connection with impervious and pervious surfaces and how Detroit has a tax on impervious surfaces. But essentially through the National Tree Benefit Calculator, which is a program within iTree, it was based on different peer-reviewed USDA Forest Service research. So they had used all of their previous research to produce a program that then gave us estimates for the calculations.
Hey, Grace, I have another question for you. So was there a difference in efficiency based on TEH density, maybe tree density of a given stand of trees?
Efficiency, are you referring to the energy efficiency?
Elizabeth, do you want to come on mic and explain a little bit?
Yeah, I was just wondering if, you know, a single tree versus a stand of trees, if you saw any difference in the value that they offered.
I did not get a chance to analyze that aspect, but with the mapping through GIS, you can do an analysis that's called a hotspot. And so you would be able to, in further studies, look at where those hotspots are, and if you see an efficiency increase given the density of the trees in that area. So this is kind of the mapping portion of it, and gathering all the information on those trees will be helpful to be able to produce future reports on things like that.
Okay, thank you, Grace. So the next question is going to Tristan. Tristan, do you think there are significant microplastics that pass your primary filter?
That question, I know that there have been other groups that have found microplastics, especially in Lake Superior. So, and microplastics definitely do pass through major systems such as rivers and stuff like that and collect and move down. So I do believe there are a significant amount of microplastics that pass through this area, and with how we're collecting it, we have noted that there are also significant amount of microplastics. Yeah.
Thanks, Tristan. So we have some time left for these 3 interns for questions. So if you have any questions, you can put them in the chat or raise your hand. And if not, we can move on to asking questions for all the interns too. So if you have any questions for any of the past interns that you didn't get to ask before, you can ask them right now.
And we're getting right toward the end of our time. It's almost 2 o'clock. So maybe just one more question before we just move on. And I appreciate there's been some really great questions so far. So that's been really good. And this, all the interns have done a great job of answering those questions as well.
I see we have a question for Cynthia. So Cynthia, are there ideas floating around for solutions that could be combined with bioswales to reduce bacterial loads?
Well, thank you for the question. I know that personally, Dr. Carmona and I have been wanting to speak to green space infrastructure engineers and kind of let them know of this issue, because it seems like bioswales overall have not really been monitored. And so just maybe letting them know and speaking to them about the levels that we're seeing and future collaborations with them would be an option. So we're hoping for that to happen.
So I think we need to move on because I know probably many people have something to do at 2. Just a little bit about the reporting. So for those people who shared their email addresses with us, we'll be getting back to you with a feedback form, so you can give us feedback on the symposium, how it was set up, and any ideas you have to improve it going forward. The interns, you should expect to hear from Emily again about your final report for the project, which is also a Google form, and that's due on August 27th. We'll also have a Google form for all the advisors and sponsors, and so we want to get your feedback as well, and that we'll send you a link to that Google form. So the recording, I just see a question about the recording. It probably will take a few days. It just seems to be that these really big presentations take a long time for Zoom to process, but once we get it processed, we'll get it up on our website. So if you go into our website and look under, I think it's under, I'm forgetting right now where it actually is, but
it's on the Michigan internship page, webpage.
Thank you. You look for the Michigan internship page and there should be a link to the recording there. And also, you know, I think our communications team, which has been on this call, has been really excited about all your projects and all your really cool pictures. And I think you can expect to hear from them. They love to get more stories from you, get pictures up on our blog, include stories from you and our newsletter. So expect to hear from them because I know that they're pretty excited about what you've been doing. And so finally, I really want to thank all of the sponsors, all of the advisors, and these amazing groups of interns. I think all of us at Michigan Sea Grant are so impressed with your projects. And I'm excited to hear that so many of you are actually going to be continuing your research into your next year. I have to say that your creativity and hard work has given me hope for the future. So good job. I also want to thank Emily. She's done such an amazing job and helping me out with this project. And so she really deserves a lot of credit for this program's success this year. So thank you so much to all of you and thank you for attending. Bye-bye.
Yeah, thank you everyone and good job interns. It was really great to see your presentations.